I want to welcome everybody here to the second of our Bampton Lectures. And I'm particularly delighted to welcome Professor Jennifer Doudna as the 2019 Bampton Lecturer at Columbia. These lectures were established through a bequest by Ada Byron Bampton Tremaine in the first half of the 20th century, and they are given at periodic intervals in the fields of religion, science, art, and medicine. Jennifer Doudna is a professor of chemistry and molecular and cell biology at Berkeley. She's renowned especially for the article in Science that she published in 2012 with her collaborator Emmanuel Charpentier, setting out a simple way of editing the DNA of any organism by using an RNA-guided protein found in bacteria. This procedure, known as CRISPR-Cas9, was immediately seen as a breakthrough and has led to multiple awards for her and to studies by her and others of the possibilities of genome editing in both non-human and human applications. Yesterday, she gave a lecture at the Medical Center focusing on the science of CRISPR and on recent work being done in the field, especially that in her lab. Most of us have read news reports and discussions of CRISPR and its possible uses and the need for caution. From the beginning, Professor Doudna has thought hard and written well, not only about the science, but also about the ethical and social implications of gene editing. Her title today is CRISPR Biology and Technology, the Future of Genome Editing. After Jennifer's talk, we'll have a response, about 20 minutes, by Hank Greely, who's a professor of law at Stanford and who specializes in ethical, legal, and social issues arising from advances in the biosciences, particularly in genetics, neuroscience, and human cells research. He has chaired or been a member of advisory councils on these matters in California and also at the NIH and the National Academy of Sciences. And then after his response, and maybe a brief response with Jennifer, then we'll open it up for questions from the audience. Professor Doudna.
Good morning, everyone. It's a great pleasure to be here in this wonderful venue and to have a chance to talk with all of you about something very exciting that's happening in biological research that extends far beyond what the science initially suggested to us in the laboratory. And what I want to talk to you about today, what we'll both talk to you about, is the awesome power of a technology for editing genomes, meaning changing the DNA in cells precisely and accurately and simply in ways that allow, in principle, correction of disease-causing mutations and all sorts of other manipulations of the code of life. And what I want to talk about, I want to start by telling you a little bit about the science behind this and how I got involved in this as a biological researcher. And then I want to talk, for the most of the time, about where this is going and what the impacts are going to be in the near term and also farther in the future. So to start things off, I want to point out that this whole field of genome editing is really a great example of the importance of fundamental research, because it's really an area of science that began with small-scale, curiosity-driven research that was focused on studying what seemed at the time to be a pretty obscure area of biology, namely how bacteria fight viral infection. This is a picture of a bacterial cell with phage, little viruses landing on the surface, injecting their DNA. And these bacteria have very little time to defend themselves against these invaders before the invaders take over. And so that's a very strong evolutionary selection for different pathways that protect the cell from this type of infection. And it was through studying this that it became clear that many cells, many bacterial cells, have in their genomes, in their DNA, an adaptive immune system that is known as CRISPR. And I'll show you just a couple of cartoons that illustrate how this works. So here we're looking at a corner of a bacterial cell that's about to be infected by a virus. And for cells that have this CRISPR adaptive immune system, they can acquire pieces of DNA from the virus and integrate them into the genome of the cell at a place called the CRISPR locus, which is this position right here. So each of these colored boxes is a little section of viral DNA that's been inserted into the genome. So it's effectively a genetic vaccination card for the cell, a way bacteria can keep a record genetically of viruses that have infected them over time. And then what these cells do with that information is make a copy of it in the form of molecules of RNA. These contain the code from the virus, a little sequence that was stored originally in the genome, and provides an address label for the cell, a way to find those matching segments of DNA from the virus should they appear in the cell again. And these RNA address labels combine with proteins encoded by the CRISPR-associated genes over here called Cas that form protein complexes guided by these RNAs. They surveil the cell looking for matching sequences of DNA, and when they find a match, they allow the Cas proteins to destroy the DNA by cutting it up. So it's a fabulous way that bacteria can adapt to their invaders and protect themselves from future infection. Now, scientists that were studying this originally were working in the yogurt industry and in the dairy industry. They were mostly studying the kinds of bacteria that are used in the culture of milk products. And they were doing this to protect their cultures from being destroyed by phage. So it's a very practically driven sort of motivation for the science. And then there were people like me that were studying this because we were fascinated by the molecular basis for this kind of an adaptive system going on in bacteria. And what emerged over time, just over sort of a matter of a few years, is that these adaptive immune systems are quite diverse in nature. And this is a cartoon that shows the numbers of different kinds of genes that are found in different CRISPR immune systems. So each line here with the colored boxes shows you a whole set of genes that are known to be part of these pathways. And so you can see that on the top, these three systems here involve multiple genes that are required for protection of cells using the CRISPR system. And down here on the bottom are a type of CRISPR pathway where in each case, just a single large gene was found to be required to protect the cell from a virus using this addressable, adaptable immune system. And so in 2011, after my lab had been studying these types of systems with multiple proteins for a few years, we started a collaboration with a colleague, Emmanuel Charpentier, to study the function of one of these proteins down here, fascinated by the fact that there were systems in nature that seemed to be simpler than these more complicated systems with many proteins, and wondering how a single enzyme, a single protein molecule, in this case one called Cas9, could provide RNA-guided protection to cells from viruses. And so that was the question that we set out to address. And what emerged from our collaboration was data showing that this protein is, in fact, a fascinating little machine that uses its RNA guide to find and destroy viruses. And I'll show you very briefly how this works. So Cas9 is an enzyme. It's a protein with chemical activity. And it's directed by an RNA molecule, this, shown by this cartoon right here, which has a 20-letter sequence on one end that can recognize a 20-letter sequence in DNA. And when that matching interaction occurs, then the protein Cas9 is able to unwind the DNA double helix and generate a break in the helix. So it cuts the DNA at a particular place that is dictated by the binding of this RNA molecule. So it literally is a chemical cleaver in the cell. Now, there's a couple of really interesting and important things about this that I want to point out. So first of all, it's a cleaver that isn't making random breaks in DNA. It's doing this kind of cutting at a place that is dictated by this RNA. And in bacterial cells, this would be RNA coming from the CRISPR locus and containing sequences corresponding to viral DNA. So this would be a great way for the cell to find and cut viral DNA molecules. And in nature, this is a system that requires two separate kinds of RNA for the system to work. Not only this piece of RNA that provides the binding site in DNA, but also a second RNA that forms a structural handle for Cas9 to assemble. And these were some of the details that we figured out in our research. And then it was Martin Yinek, who was a postdoc in my lab working on this science, figured out that it was possible to link together these two RNA molecules in nature to form what we called a single guide RNA that would contain the address label on one end and the binding site for Cas9 on the other. And this might seem like a small change, but this was one of those experiments that, although simple to do, had profound implications. And the reason is that as soon as Martin showed that you could design single guide RNAs with different letters on this end to allow Cas9 to find and cut essentially any desired DNA molecule, we realized that this system that had started off in our lab as a curiosity-driven project was going in a very different and much more profound direction. And the reason is that this ability to dictate a cut site for DNA in cells is something that many other labs had come to appreciate could provide a powerful technology for something called genome editing. And to show you why, I just want to show you this cartoon. So in bacteria, a double-stranded break to DNA leads typically to rapid degradation of the DNA, destruction of the DNA. So it's a great way to protect cells from viruses. But in our cells, in eukaryotic cells, plants, animals, fungi, cells that have their DNA inside a nucleus, these are cells that have the ability to detect breaks like that and repair them. And so when cells experience a double-stranded DNA break, they can identify it and fix it by processes that lead to either a small disruption in the DNA sequence, often a single letter change to the DNA code, or they can insert new segments of DNA in the process of repairing the break. And so scientists over the last couple of decades had studied these repair pathways and realized that if you could introduce a double-stranded break into a genome, into a DNA molecule, a chromosome in cells, you could trigger this kind of repair and thereby alter the genetic code at a particular position in a gene. And why would you want to do that? Well, there's all sorts of interesting discoveries that have been made by understanding how genes contribute their information to the formation of a tissue or a whole organism. And so lots of classical genetic experiments have relied on random changes to DNA that scientists can study. But imagine you had a tool that you could use to make a desired alteration to particular genes at will. And that's exactly what genome editing allows scientists to do. And the amazing thing about the CRISPR-Cas9 protein is that nature has figured out how to program this single protein to cut DNA at a programmed position, a position dictated by that DNA molecule. And once we understood how that worked, we could easily imagine how you could use it as a technology in human and plant and other kinds of cells to make desired alterations to genomes. I'll show you a little video that I showed this yesterday for those of you that were at the talk, but this is really just a kind of an artist's rendition of what we imagine this kind of editing looks like in a cell. So we're zooming into a eukaryotic cell where the DNA is inside the nucleus. And not only that, the DNA is compacted in the form of chromatin. So it's wound around proteins to make it fit inside the nucleus. And this bacterial Cas9 protein is able to search through all of the DNA in the nucleus to find a sequence that matches the sequence of the guide RNA that it contains. And when it finds that match, it opens the DNA, cuts it, and then hands off those broken ends to repair enzymes in the cell that can fix the break by doing things like making a small change, or in this example, inserting a whole new segment of genetic material at the position of the break. So you can imagine that when Emmanuel Charpentier and I published this work in the summer of 2012, there was a lot of excitement in the field about the potential of this technology to help scientists do the kinds of genetic manipulations that many had dreamed of doing, but up until then had been very difficult to do with either just stochastic changes that happen in the DNA of cells, or by using earlier technologies that, while they could allow these changes to occur, were difficult to use and required a lot of expertise on the part of scientists. So what happened next, I think, was beyond what any of us could have imagined in the beginning. I took this slide from a publisher, Elsevier, that publishes a number of scientific journals, and they just plotted out the scientific publications that came from earlier technologies for genome editing that involved using engineered proteins that could find and cut a particular DNA sequence, but required new proteins to be engineered for every experiment, every different cleavage site or editing site in a cell. And that's these technologies over here, shown by the blue and sort of gray colors. And then here's what happened when the CRISPR technologies was introduced. And you can see that the field just began to take off. And really, the number of publications in the scientific literature has literally been exponential in this field since then, increasing every single year exponentially, as more and more labs globally have adopted this technology for different kinds of genome editing applications. Now, I'm a scientist that, I've always done biochemistry and structural biology in my lab. So we're a lab that asks the how questions. How do things work? And I wanted to show you just briefly a little bit of what we understand about how this amazing bacterial system is able to find a particular place in DNA, make a cut, and then hand it off for repair in animal and plant cells. And so this is one of the questions that we've been asking ourselves in the lab over the last few years, is what is the mechanism of DNA recognition? So we know that it involves this pairing of the letters in the RNA molecule here and the DNA, but we also know that it requires DNA unwinding, that literally the protein has to be able to separate the DNA strands of the DNA double helix. And this process, I'll show you a picture here. This is actually a photograph of a 3D printed model of Cas9. So the protein is shown in white. It's holding on to the guide RNA, which is the orange molecule, and a target sequence of DNA in blue. So you can see the double helix of DNA winding its way through the protein. And as it passes through, it's actually, this is a target sequence in DNA that is interacting with the RNA to form an RNA-DNA helix inside the protein with the other strand of DNA separated away. And that allows the protein to get in there and get access to both strands of DNA and make a cut. So these enzymes that work like this are able to find a sequence of DNA even in the context of a very large genome. For example, the 3 billion base pairs of the human genome. This is an enzyme that can find a particular position and make a cut. So it has to be pretty good at figuring out when it's in fact engaged on a bona fide target DNA sequence. And we're still studying how that works, and it doesn't get it right every single time, but it's pretty good. It's pretty accurate. And this is a slide that just shows another view of a structure of one of these types of proteins. So you can see that we've got a number of different colored parts of the enzyme, this protein, that are holding on to both the guide RNA and a molecule of DNA. And there's a nice cleft in the center that is really the business part of this enzyme. It's the part that is able to pry apart those DNA strands and interact with the RNA. And as scientists, one of the things that we're fascinated by is that this process of opening up the DNA at a particular place and positioning those strands for cutting is a process that happens without an external energy source. So imagine that. This is an enzyme that finds a place in a chromosome that matches its guide RNA and opens up the DNA, but it doesn't have any way of, it doesn't have an external source of energy to do that. So we know it has something to do with the structural interactions that this protein makes with DNA. And I have a video here that just shows you what we call a little morph between the different structural states of this Cas9 enzyme as it binds to RNA and to DNA. So it starts off with the protein in, oops, sorry about that. It starts off with the protein in the pre-bound state, morphing to the structure that it forms when it binds the guide RNA. So there's a big rearrangement of the structure that opens up a channel in the center of the protein where this guide strand of RNA can interact with a DNA molecule. So this thing searches the cell, and when it finds a matching DNA molecule, it binds and triggers additional structural changes in the protein to accommodate that RNA-DNA helix. And then finally, a chemical cleaver in the enzyme right here is able to swing into position to actually cut the DNA strand. So when you look at something like this, you get a sense of the way this molecule really works as a little machine. It's really designed to interact with DNA at a precise place and trigger cutting only when it's engaged on a target sequence that matches its guide RNA. And that's very important for how it's been able to be deployed as a technology for all sorts of research applications, but in the future, increasingly, applications that are going to solve real-world problems and affect, I think, all of our lives going forward. And so what I want to do now is I want to turn to some of the ways that this technology is being utilized. And I want to talk to you in particular about 3 areas where I think there are both exciting applications, but also potentially very profound ethical challenges. And that's something that maybe we'll get into with Professor Greely. So these are in areas of public health, in agriculture, and biomedicine. So first of all, one of the very first things that people started to think about when genome engineering, as it used to be called, first came on the scene, and this was already going back a decade at least, was the possibility of being able to correct disease-causing mutations in people. So imagine that you had the ability to change the DNA precisely in the tissues of a patient affected with a genetic disease. And I'm going to give you one example of this, and that's the example of sickle cell anemia. So this is a blood disorder that's been well studied for a long time. It's caused by a single mistake in the DNA that's shown right here. Here's the sequence of DNA in a normal gene, and then here's what happens when the gene is mutated at a single position. So we have a single base pair change compared to the normal cell that gives rise to a mutated form of hemoglobin, which is the predominant protein made in blood cells. And when this mutated form of the protein is produced, it generates cells that have a very classical kind of sickled shape. And when they get into the bloodstream, they don't fit very well, especially in very tiny capillaries. And they can get stuck and cause severe pain and other problems for people affected with this disorder. Now, there's some interesting biology here, because it turns out that in parts of the world where malaria is endemic, it can be protective for people to have one mutated copy of this gene that actually helps prevent them from being infected by malaria. But if they were to inherit 2 copies from parents that donate 2 of these mutated copies, then they actually have sickle cell disease, where their cells are sickled and they go through terrible cycles of pain due to this kind of disruption. And so this is the kind of system where we know very well what the cause is of this disorder. It's very difficult to treat these patients, and they require blood transfusions and other kinds of really palliative treatments. But imagine you had a way to go in and correct that disease-causing mutation. And that's one of the areas where I think genome editing is likely to have its earliest impact clinically, because of the ability, in principle, to correct these disease-causing mutations in blood cells. And that's a type of genome editing that we refer to as somatic cell genome editing. It's a type of application of this technology that will affect an individual, but it will not affect their kids, right? So there's no question about this being a trait passed along to the future. It's a very much analogous to any kind of traditional therapy that you would use on an individual to help them treat their disorder. Now, other ways that I see public health being impacted by genome editing, I want to point out a couple of things. One is that it's increasingly possible to use genome editing in animals. And one of the interesting examples of this that has potential impact in human health is the use of genome editing to alter pig genomes to make these animals better donors for organs. And so in recent experiments, both in academic labs and in companies, it's been possible to do two things with genome editing. One is to remove all of the endogenous viral sequences that are part of the natural pig genome that could be problematic if they were transplanted, their organs were transplanted into humans. And the second thing is to change their DNA so that the resulting organs are more human-like, that they're more likely to be accepted by a human recipient of these organs. And this is a project that is going forward very quickly, given the ability to manipulate pig genomes in a way that just a few years ago was basically not possible. Now, another area of potentially very interesting and large impact on public health, but also with, I think, important ethical challenges to it, is the use of genome editing for something called gene drives. There's been a lot of discussion about this in the media, and basically the idea here is that when you have a genome editor that allows rapid changes to be made to a genome, there are ways of using it that will drive a genetic trait through a population very quickly. And this typically would be done in a population that's of rapidly reproducing organisms, like mosquitoes, for example. And this is a slide that I took from the magazine Science News. So they had a nice diagram here that shows you how gene drives work. So on the left-hand side, we're looking at, this is how a normal inheritance happens with classical Mendelian genetics, where a trait would be passed along to just some individuals in a population according to this pattern. But in a gene drive, what happens is that the inheritance becomes non-Mendelian, and there can be very rapid spread of a trait through a population by the activity of the genome editor that gets inherited along with the trait. And so you can see that very quickly this altered gene is spread through this population and becomes intrinsic to the genomes of virtually all of the animals eventually in just a few generations. There's lots of interest in using this as a way to limit the spread of mosquito-borne diseases, but there's also the potential for environmental damage, and that's something that scientists right now are really grappling with. How do we balance the important opportunities to prevent the spread of disease, which could be incredibly positive, have a great impact globally, against how do we balance that against the potential risk of releasing something into the environment that can't be controlled once it's released. So then I want to just mention also that although there's a lot of attention focused right now on public health and biomedical uses of genome editing, I personally think that in the near term, the bigger impacts are going to be seen in agriculture. And the reason is that there are now incredibly interesting opportunities to use genome editing in plants that will alter the way these organisms in our environment and that we use agriculturally can be controlled. This is one example that is taken from the work of Zach Lippman, who's here at Cold Spring Harbor Labs, not too far away. And he was able to show, this is a paper published now about 2 years ago, that you could use genome editing in tomatoes to control the number of fruits that they produce. And the amazing thing about this is that this is a gene that is found in many different kinds of crops. And so you could imagine having a way to alter the yields of crops without changing any other characteristics that they have. Traditional plant breeding requires introducing random changes into plant genomes, selecting over many generations plants that have the desired traits and not undesired traits, and not being able to control the changes to DNA that happen in the process. And here we have a precise tool that allows alteration of just a desired gene and nothing else. Another example of this is work that was done in another academic setting. This was work done at Penn State University to change a single gene in mushrooms that controls browning of these mushrooms when they're cut. And this type of editing, like the alterations that were made in tomato, doesn't change anything about doesn't introduce any foreign DNA into the plant genome. It just alters a single gene that's there naturally. And so there's been a lot of discussion about whether this kind of alteration to plants should be considered something, in the category of genetically modified organisms, GMO, which is sort of become really a hot button issue in many parts of the world. And this work, actually with the mushrooms, led to the discussion at the US Department of Agriculture about this question and the decision to, in the US, not regulate plants that are altered in this way. So these are plants that don't have foreign DNA introduced into their genomes and are therefore not considered to be GMO here in the US. And interestingly, in Europe, the very same types of agricultural products are considered GMO because they are products that have come from genetic manipulation in a laboratory, even though the DNA that results doesn't have foreign DNA inserted into it. And so this has really sparked a fascinating discussion that's very much ongoing about how do we regulate these kinds of products. They're coming down the pike very quickly because more and more companies are able to use this kind of technology to make alterations to plant products that all of us will eventually be able to get access to in our supermarkets. And we have to decide as a society and then sort of globally how these are going to be regulated. How do we decide what's safe and how to control it? And I hope maybe we'll come back to discussing that at the end. And then finally, I just want to mention one other area that I think may not have come to your attention if you're watching what's happening with genome editing, and that is that it turns out that these enzymes that are part of CRISPR pathways are quite diverse in their chemical activities. And recently, it's emerged that some of these proteins can be used to detect DNA and RNA and trigger a chemical reaction that can be easily detected in a point-of-care setting, meaning that you don't need fancy equipment, you don't need a lot of time. You can use these enzymes to tell you whether the DNA of a virus or a bacterial cell is present in a patient sample, and therefore have the opportunity in principle to have very simple ways of detecting infections, maybe even doing things like detecting genetic variation in a person. We used it recently in our lab in a kind of a blind test to figure out who had blue eyes and who had brown eyes in the lab, just by having students spit in a tube and using this diagnostic approach. So this also, I think, is both very exciting, but also raises interesting ethical questions. What if you could go to a Walgreens and buy something like this for a few dollars, and you could figure out if you had a mutation in a gene that is very likely to lead to breast cancer? How do we think about that? How do we give people enough information that they can make decisions about their health care and do that in a responsible fashion? So finally now, just in the last couple of minutes, I'd like to turn to a topic that has gotten a lot of attention in the media, and it's this subject of what we call germline editing in humans. And to explain this, I have to point out, so as I mentioned earlier, a lot of the work, the vast majority right now that's going on in biomedical applications of genome editing focuses on somatic cell editing. And that means making changes to DNA in an individual that are not inherited by future generations. But germline editing is different. That means making changes in eggs or sperm or embryos such that those genetic changes become part of the individual and they are passed on to future generations. So this is a much more profound way of doing editing because if you think about it, it's really allowing humans to alter human evolution. We can alter ourselves at the level of our DNA and not do it randomly, but do it in a controlled fashion. This is a picture that shows how sort of one way that this is done. This is actually this is not a human embryo. This is a mouse embryo that's being held by a pipette on the left with a needle injecting the CRISPR-Cas9 molecules on the right. And it turns out that this procedure is not very hard to do for people that know how to work with these kinds of embryos. And in fact, one of the very first applications of CRISPR-Cas9 that was published in the scientific literature after Emmanuel and I published our work was research using this technology in mouse embryos to make changes to mouse DNA that are inherited by those mice. And this is now done very widely in many animal models that are used to study diseases in laboratories. But of course, you could imagine that this could be also used in principle in human embryos. And that's something that I started to think about a lot back in around 2014 when it was clear that the research using genome editing was advancing at a very rapid pace and at a time when virtually nobody outside of the scientific community, including our governments, our regulatory agencies, our legal teams, bioethicists, had any idea what was coming down the pike and what was bubbling up out of research laboratories. And that made me very uncomfortable. And so I convened a meeting in early 2015 that Hank Greely attended, and we discussed this topic. And that led to several articles that were written and, importantly, a series of meetings that were convened by the National Academies of Science here in the US and in cooperation with other countries, ultimately leading to publication of this report that appeared in the spring of 2017 about human genome editing, and in particular, investigating this question of human germline editing. And in this report, if you read it, which you can download if you want to, it basically describes where the technology stood at that time and made a recommendation to the global scientific and clinical communities that there not be any use clinically of human germline editing until the technology and the societal and ethical implications of its use in the germline could be carefully analyzed and frankly, publicly discussed. But unfortunately, that did not stop this procedure from being used in the clinic by this gentleman, He Jiankui. This is a picture I took of him presenting his work in Hong Kong last November, where he told the convened audience of several hundred people who were at a meeting to discuss human genome editing that he had, in fact, utilized CRISPR-Cas9 in human embryos. Those embryos had been implanted and resulted in the birth of twin girls that had alterations to their DNA. Now, the stated purpose of that work was to protect these girls from HIV infection. But as the data were being presented at this meeting, I think I think virtually everyone else there had the same sort of creeping feeling that the work had been done in a completely unethical and medically indefensible fashion. And I just want to show you one slide to illustrate what I mean by that. So this is a slide that shows the actual changes that were made to the DNA of these twin girls. And you don't have to look at the details. I just want you to notice that lines 3, 4, and 5 here don't look like these. And so what this is showing you is a cartoon of the DNA in a gene called CCR5 in people that we all have. We all have this gene, and it's been found to be what's called a co-receptor for HIV. It's a gene that produces a protein necessary for HIV to infect human cells. And in very rare individuals in the natural human population, there's a deletion of this gene that's cartooned right here called delta 32 that renders the gene non-functional. It doesn't seem to have a deleterious effect on those people. And in fact, what it does is to protect them from HIV infection. So there's been lots of interest in this in the medical community over the years. And so what He Jiankui did was to use genome editing to alter the CCR5 gene in these 2 embryos. And he introduced changes that are shown down here. And you just need to notice that none of these 3 lines, which represent the changes in those human embryos, look like this. So what he had done was to introduce changes in the CCR5 gene that, to our knowledge, have never been seen in the human population. And not only that, have never been tested in animals. So he was truly doing research on people that had never been even tested before in animals. So that's, I think, very, very shocking and really completely unethical. And secondly, it became clear that the way that the patients, the parents that were giving birth to these kids, had been informed about the technology and the way their consent had been obtained, as well as any effort to figure out how you would follow the health of these girls after their birth, had been completely bungled. So this raises a very big question. Where do we go from here? What do we do? Is the future this, where we're going to have CRISPR babies and people going to IVF clinics and hand in a menu and they can check off boxes for the kinds of genetic features they want for their children? Well, I don't think this is coming anytime soon. And it turns out that a big impediment to this, aside from anything else, is that we don't understand enough about the human genome today to be able to make changes that would give rise to these kinds of phenotypes for the most part. And many of the features that we all have are the result of many, many genes that interact. And for the most part, we don't know what those interactions are like. But there's no doubt that over time, a lot of information about those pathways is going to come forward, partly because of the use of CRISPR-Cas9 and related enzymes in research studies. And there's tremendous interest in the potential of human germline editing to do things that we might consider to be very interesting medically and very beneficial, such as removing a disease-causing mutation from a family so that they don't have to be afflicted with cystic fibrosis or muscular dystrophy, things like that. So there's no doubt in my mind that this is technology that is coming and it will be used in the future in this fashion. The question is how, and how do we control its use in a responsible way? So one thing that's happening currently is the work that's being put forward by different international organizations to convene groups of scientists that will deeply dive into the technical and ethical and societal questions around human germline editing to come up with not just guidelines, but in fact, very detailed restrictions and requirements for anyone that in the future wants to use this technology in the clinic. And the hope is that these restrictions and requirements will form the basis for regulatory guidelines that are put in place by governments around the world. So the World Health Organization has announced their committee, and very soon there will be an announcement from the national academies of a related committee, and the hope is that these teams will be able to work together to coordinate a response to this event that happened last November. One of the challenges is just understanding how we approach the issue. So, one of the first things that this World Health Organization panel proposed was having a registry for all human CRISPR-related experiments, which might sound like a good idea, except that when you think about it, there's so many experiments that are going on mostly in somatic cells, meaning non-heritable forms of gene editing in humans, that this is probably not realistic. And so right now, there's, again, a lot of discussion about how this call might be modified so that it's focusing on just those folks that might be interested in using genome editing in embryos and for the purpose of eventually using it clinically. And that's it. So I just really wanted to conclude by pointing out that the technology continues to advance very rapidly. This ability of proteins to use an RNA guide to find a position in the DNA of a cell and make an alteration to it is going to, in my mind, continue to be a very powerful tool for the foreseeable future. And there's lots of research going on here and at many other places that are uncovering new and interesting activities of these proteins that will continue to advance this whole area of scientific research. We know that delivery is a big challenge. How we get genome editors into cells and tissues will continue to be a sort of a bottleneck, I think, for using this technology in patients, in certainly in somatic cells, as well as control, how we ensure that the kinds of changes that happen to DNA after interacting with CRISPR-Cas enzymes are the ones that we want. And control also refers to the ability to responsibly use and regulate this technology. And finally, fundamental research will continue to drive forward all of these areas of science. And I do want to just finally, again, emphasize the importance of fundamental research to advancing these areas of science and technology. I'm grateful to my research group. All of us, so I'm here giving talks, but back in the lab at Berkeley, these folks are actually the ones who are doing the experiments. And I'm incredibly grateful to my team of people that are creative and doing work that is going to drive the field forward. We've also benefited from great collaborations, increasingly with clinical teams at UC San Francisco, our neighbor university across the bay. And finally, we require funding from public and private agencies that provide the money to do the research that we do. And in my own case, I'm particularly grateful to the National Science Foundation, which is a taxpayer-funded organization here in the US that sponsors research that's mostly not medically focused. And in fact, they gave us a small grant in my very first couple of years working on CRISPR biology that allowed us to do the exploratory science that eventually led to working on CRISPR-Cas9. So thank you very much, and for inviting me.
Good afternoon. I hope no one came up here expecting a confrontation between Jennifer and me. This is a much more complimentary than confrontational talk. I first met Jennifer in January of 2015 at Napa. We've probably run across each other a dozen times since then or so. At the time I met her in Napa, it was already clear that CRISPR was the most important biological discovery of the 21st century, at least to date. It's not so important because it's the first way to gene edit. Gene editing, as her slide with talins and zinc finger nucleases pointed out, gene editing has been around for a while. I'd even extend that back farther to my Stanford colleague Paul Berg and recombinant DNA almost 50 years ago. So it's not that CRISPR is the first gene editing, and it's not going to be the last one. Somebody's going to come up with new things. It's already bubbling into CRISPR-Cas 147 and all these different CRISPR things, but it's the Model T. There were cars before the Model T. They were really expensive, and they broke down all the time. So if you wanted to have a car, you had to be rich, and you had to have a full-time chauffeur mechanic. And for those of you who get this cultural reference, you had to hope that he wasn't a charming Irishman who ran off with your daughter. Downton Abbey folks are not big in this group. Okay. Anyway, Henry Ford came along, and the model, and so there were cars in California. There were maybe 1,000 cars in California. There are now 40 million people in California and about 60 million cars. All of us Californians are centaurs, we're half human, half car. What changed California wasn't the car, it was the Model T. It was an effective, reliable, cheap car that everybody could have, and pretty soon everybody did. And what's revolutionary about CRISPR isn't gene editing. It's gene editing that's cheap, effective, accurate, easy, that everybody can have. And everybody is. And so even over 4 years ago, it was clear that CRISPR was an enormously important invention. It was also clear that Jennifer didn't invent CRISPR. Unnamed bacteria, billions and billions of years ago, invented CRISPR. But Jennifer and her colleagues, aided and abetted by people at other places as well, and other groups, invented the tool, invented CRISPR as a human tool. And that's what was really important. But what I was really impressed with in January of 15 at Napa, this meeting, which was the result, in some respects, of, as she has recounted in other contexts, one of Jennifer's dreams, she was very concerned, very sincerely concerned about the ethical consequences and the social implications of CRISPR. So I have been impressed watching her over the last 4 years, just purely as a law professor, professionally. One of the things that's impressed me is she's gone from a very good scientific presenter to an excellent public speaker, two things that aren't as closely related as they might otherwise seem. So I have professional compliments for you there. But I've been impressed watching her continue to move the science forward and continue to talk about and be concerned about and be involved with the ethical issues. So my talk here is not just complimentary to Jennifer's, but it is complementary to Jennifer. I want to do two things. First, I want to talk about what I think are the most important ways to think about CRISPR in humans. And second, I want to talk about the He Jiankui fiasco. This is one area where I do differ with Jennifer. She called him a gentleman. I would not call him a gentleman. But I'll get into that more a little bit later. First, though, I actually think the most important consequences of CRISPR during our lifetimes, and maybe forever, will not be the human uses, but will be the non-human uses. And yet, I don't think this audience would be so big if this were just about non-human uses of CRISPR. We humans, for some odd parochial reason, seem to care more about humans than we do about mosquitoes and fruit flies and other things. So although I'm going to focus on humans, because most of the discussion has focused on humans, I do want you to remember that the little bit that Jennifer talked about with non-humans, that's probably 90% of the important consequences of CRISPR in the next 20 years or more. But we care about people, so let's talk about people. I think thinking about the ethical issues raised by CRISPR in people is really a two-dimensional task. There are two directions, two axes, in which most of the controversy has landed. One axis is somatic versus germline. It is things that change the body cell, the cells of the soma of the body, but don't get passed on to the descendants, and things that do get passed on or may get passed on to the descendants because they change not just the somatic cells, but the germ cells, eggs and sperm, or the precursor cells that give rise to eggs and sperm. And so that's one of the dimensions. Is it somatic? Is it germline? The other big dimension is medical versus enhancement. Is it for improving health, or is it for making super babies, or X-Men, or enhancing people in any way that your fantasies would lead you to? Now, I want to note importantly here, that one in particular is a fuzzy line. Enhancement versus therapy is not always obvious. I have progressive bifocals because I'm old and I need reading glasses. Getting reading glasses, is that a treatment? It takes me back to where my eyes were 20 years ago. Or is it an enhancement because somebody my age normally needs reading glasses? That's just a normal function of human life. Vaccines. Vaccines prevent disease, but they do it by enhancing our immune system. So the enhancement-treatment dichotomy isn't clean. But that's not going to be too important. I just want to make sure that's noted. So germline, non-germline. Germline, somatic. And enhancement, non-enhancement. You can think of this as a 2x2 matrix, so you got 4 boxes. First box is somatic, medical. And this one has almost no new interesting ethical issues. This is using CRISPR to cure people of sickle cell anemia or beta thalassemia or cystic fibrosis. That's not to say it has no ethical issues. It has lots of ethical issues, just as all new and expensive medical therapies have ethical issues. There are questions of safety, there are questions of access, there are questions of how you figure out fairly which diseases to go after first. There are questions when new gene therapies are priced at $1.3 million per patient. There are lots of ethical questions, but there's nothing about it that's special to CRISPR. And that's important, and that's the area CRISPR is going to make its biggest impact in humans, is going to be in somatic cell treatments. The germline is where all the fighting's been, as Jennifer pointed out. And I am of I'm of two views on this. I have this, I don't know whether it's genetic or environmental or both, but my personality is such that on any controversial issue, I like to move to the middle so I can get shot at from both sides. That way I don't have to worry which direction the arrows are coming from. They're coming from everywhere. On the one hand, the human germline genome, in my best bass voice, is not the Ark of the Covenant. It is not the essence of our humanity. It is not what defines us as a species. It is not a sacred relic brought down from your favorite mountain that we need to cherish and preserve. It is a constantly changing flux. What is the human germline genome? Well, there's 7.5 billion of us humans. The human germline genome obviously is mine, but some of you may disagree. There's 7.5 billion human germline genomes, and they change every generation because of mutations that happen during our lifetimes to our eggs and to our sperm. What we pass on to our kids isn't exactly what our parents passed on to us, and so on down the line. Not only does it change, that's a random change, but non-trivial. Not only does it change with each generation, but human activity directs the change in some respects. So for example, our genes have different variations or different numbers of variations than our hunter-gatherer ancestors from 20,000 years ago, because agriculture changed our genome. We have more genes to digest starches than our ancestors did. We have more immune system genes because living in farming communities meant we had more diseases to have to deal with, which changed our genes. And medicine changes our genome. It was really not quite 100 years ago when two Canadians discovered that you could use insulin to keep people with type 1 diabetes alive. Now, type 1 diabetes, the kind where you have to inject yourself with insulin every day, is not exactly a genetic disease, but it's not exactly a non-genetic disease. There are variations in something called the human leukocyte antigen system. It's an immune system set of genes that greatly predispose some people to get type 1 diabetes. It's also known as juvenile onset diabetes. So people with type 1 diabetes didn't pass it on to their kids because they didn't have kids for the most part, because they died when they were 8 or 10 or 12. Insulin, Canadian intervention, Canadian meddling, comes up with insulin. Suddenly these people can live long enough to have children. And so, although I've never seen the census done, I'm quite confident a higher percentage of people in America today have genes that predispose them to type 1 diabetes than did 90 years ago, because those people, even when they get diabetes, are able to reproduce and have children who inherit those variations. Our environment, our culture, our society, our technologies direct changes in our germline genome. So I don't see the germline genome as a sacred object that needs to be preserved. It's a thing. It's a thing that changes. Maybe we can make it better. Maybe we can make it worse. It changes randomly. Will our trying to change it intentionally be better or worse than our trying to change than it changing randomly? The answer to that, I'm sure, is yes. It will be better and it will be worse. But not necessarily. I don't know where the balance is. So I'm not with the group that says no germline genome changes ever. On the other hand, I don't think it's very useful. Almost everything medically that you'd want to do with germline modification, you can do with other methods, particularly one called pre-implantation genetic diagnosis, where you can take embryos, take a few cells from the embryo, test those cells genetically and say, this embryo is going to get cystic fibrosis. This embryo won't. This embryo is a carrier. This embryo isn't. And then only transfer to the woman who's going to be the mother the embryos that are safe. How many of you have heard of PGD, pre-implantation genetic diagnosis, before? This is a good crowd, about a third. Usually I only get about a 15th. Well, no, about 15%. I get about a sixth normally. Most people think it's science fiction. The oldest human born from PGD is now 28 years old, maybe 29, born in 1990. This is clinically available for the last year for which we had data in the US, 2016. Probably 15,000 kids were born after PGD, and none of them ended up missing an arm, a leg, a head, or any other appendage. It's a safe, IVF clinical process. If you can do PGD, you don't need to change an embryo's genes to make sure it doesn't have cystic fibrosis. You just make several embryos and you pick the ones that will be safe. In 2016, I published a book with the scary title, The End of Sex. It's not as scary as it sounds, and the future of human reproduction, which predicts a massive increase in the use of PGD because I think we're going to start being able to make eggs and sperm from skin cells. So if you've got PGD, why do you need medically to do germline treatment? There are there's one really good reason and one not so good reason. The really good reason is sometimes there are couples who desperately want to have genetic children. Now, one can argue about whether people should desperately want to have genetic children or not. But people do. Many people, not everybody, but lots of people do. But some of them would have to have genome editing in order to have a healthy kid. If you've got a couple where both members of the couple have a genetic disease, like, say, cystic fibrosis, they cannot make from their own genes a child without cystic fibrosis because the only genetic variations of the CFTR gene they have to give are the disease variations. If you've got somebody who's got 2 bad copies of a gene for a dominant disease, where it only takes 1 bad copy to confer the disease, something like Huntington's disease, they can't have a healthy kid from their own genes because any of their children necessarily will inherit at least 1 bad copy of the gene, and with a dominant disease, 1 bad copy is enough. How many couples like that are there in the world? I don't know, probably not more than a few thousand out of 7.5 billion people. But should we discriminate against them because of these germline fears? So I think the germline issues are overblown. I'm going to talk briefly, very briefly, really briefly, about the enhancement issues. It's okay. About the enhancement issues. I think these are real issues. They're complicated issues. I just flew in from someplace else last night, but my arms are not tired. This is not my natural fur. This is not my unamplified voice. This is not a cave. Nothing about our lives is natural. It's enhanced. What humans do is enhance themselves and their lives. Is it different to do it genetically than non-genetically? Maybe. Is it different to do it germline versus non-germline? Well, maybe, although we pass on a lot of things to our kids through things other than genes. Yankee lover, Yankee hater, that runs in families, and it's not genetic. So the enhancement issue is a real one, and we see it in sports with performance-enhancing drugs. What should we allow? What shouldn't we allow? I think that's a big societal debate that we need to have. I tend to be on the pro-enhancement side as long as there's safety, no coercion, and fairness at how it's distributed, big ifs. But I think it's a debate we have to have, but it's a debate that's not just about CRISPR. It's a debate that's about devices. It's a debate that's about drugs. It's a debate about, the supercomputers we carry in our pockets that we call phones. I remember when calculators first became usable, and there were huge debates about whether students should be able to use calculators in their math classes. Those are all enhancements. And those enhancements change us. So, enhancements versus therapies, one axis. Germline versus somatic, another axis. Those are the big issues with respect to CRISPR's application to humans. Maybe in the discussion we can talk a little bit more about her. I'll just say that that is one of the worst experiments I have ever seen, without regard to the fact that it had anything to do with germline, but that science needed to react a little bit differently. We need deterrence. He should rot in jail forever so nobody else thinks this is a good idea. We need disclosure so the people who found out about it early would know that they should tell somebody and would have a good place to go. And we need a little bit more humility by science. In the early reports, they all said, listen to society, interact with society, talk to society. After the Hong Kong summit, we didn't hear that so much. I don't think it was really intentional, but that sounded more like, well, we shouldn't do anything until we scientists think that it's right. It's not an issue for scientists. Well, it's an issue for scientists, but it's an issue for scientists and everybody else too. And I think scientists know that. We just need to be a little more open, a little more humble about the fact that science is part of society and ultimately is and must be governed by society. Thank you for your time and attention. Let's talk.
I don't know if you want to respond for a minute back and forth, and we'll open up the questions from the audience. Either way.
Berkeley, I like it.
Okay.
More about Berkeley.
Shall I open to the audience, or do you want, Jennifer, do you want to respond to something?
I'd love to open it up to the audience. Sure. I guess I would just say that I think Hank Greely and I are largely in agreement. The thing that I've become more aware of as I've learned more about human in vitro fertilization and the procedures that are associated with it, such as pre-implantation genetic diagnosis, as he mentioned, is that it's not necessarily as easy or trivial to do that kind of analysis as you might imagine. And I'm sure that technology will continue improving. But I do think that there's kind of these two parallel tracks that are going on right now, where there's advances in the IVF and PGD technologies in clinics, and that's sort of going in parallel with what's happening with genome editing and the ability to manipulate embryos. Right now, one of the reactions from a very prominent scientist who was with us in Hong Kong after the announcement by He Jiankui was that what his work showed was that it's relatively easy to do human embryo editing. It's very difficult to do it well right now. That's the way it is today. But that will change over time. And so I do think that we have to really kind of be paying attention to what's happening both technically with advances in vitro fertilization technology and advances in human genome editing, because at some level they're going to be, they're going to run alongside each other, and each provides, different opportunities but challenges as well for how we're going to be utilizing them in the future.
Yeah, we don't even disagree that much about that, because I do think when something's new and risky, you do it on the places where there's the most benefit. You do it on diseases, you do it on the worst diseases. You don't do it to try to prevent a chance that somebody's going to get HIV 20 years later. But as it gets more regularized, more accurate, safer, and we have more confidence in it, then maybe you move to doing gene editing so that you don't have to do PGD every generation. And you can save future generations from having to worry about, did the embryo get my CF gene or did it not? So in the long run, I think that's right. But the really compelling use, and that's a useful use of germline, but I think the compelling uses are relatively few, certainly in the near term.
Agreed.
Questions? Sure.
You want them to come to the microphone?
Yeah, let's see. Do we have him?
You've got a couple
Yeah,
mics.
come up to the microphone. Yep.
Hello, John Morris, New York Genome Center. So thank you very much for the talk. It was very fascinating. So I guess I'd like to ask a question on the lines of perhaps maybe public policy and the availability of the reagents for these experiments, where in the context of like personal genetics testing and the phenomenon known as biohacking. So there are some viral videos going around of people buying like Cas9 proteins and just trying to like inject themselves with it. And then there's also the aspect of, with personal genetics testing, someone might, who's not educated in genetics, could see a report saying that, oh, you have like an ApoE variant that predisposes you to something like Alzheimer's. So what's to say that people who are perhaps concerned for themselves and do not have really the education or training to conduct these experiments might just go out and try to buy a Cas9 protein with guides for ApoE and just try to infect themselves?
Well, they could do that, but I don't think they'd get very far. It's not as simple as simply injecting your thigh with a protein to get genome editing to work. This is where the whole challenge of tissue delivery comes in. But you know, you raise an interesting question. When you have a technology like this, and I think it was clear in these talks, but maybe I'll just say it explicitly, but that's that, you know, one of the wonderful but also very challenging things about CRISPR genome editing is that it's widely available, it's easily accessible, and it's not that difficult to use. And so, you know, I think your question really speaks to that challenge of how do we control the, you know, the access that people might have who want to do something that, you know, could be considered either, you know, at best unsafe or ineffective, at worst unethical and dangerous from using it. And to me, that's, again, it's not unique to CRISPR. It's really a property of any technology that is widely available. We could apply that same question to asking ourselves about software engineering, or, you know, hacking computer programs, and lots of other things. So I think it's just the general question that we face in our society today, where technologies are moving forward incredibly quickly. They're very enabling in many cases, and yet it's relatively difficult to regulate their use.
You know, it's funny how the vocabularies are shifting between the silicon-based life forms and the carbon-based life forms. Hacking is moving from computers to biology. Viruses moved from biology to computers. There isn't a relationship. The best example of what you're talking about is another fellow resident of ours of the Bay Area, a guy named Josiah Zahner, who is not as crazy as he sometimes seems, who is the one who injected himself. He will sell you a do-it-yourself CRISPR kit. Now, it's to change yeast, and that's relatively easy. He's now working on selling you something about frogs, which is trickier. He's a relatively not mentally ill adult. If he wants to do this to himself, it's actually technically against the law. It's a violation of the Federal Food, Drug, and Cosmetic Act, but I don't care, and the FDA doesn't really care. But if it gets out widespread and people do it as if Gwyneth Paltrow tells people to do it and so they all do it, that's a significant public health issue at this point. I do think, and similarly with genetic testing, there will be people who get a negative test result from 23andMe about BRCA1 and BRCA2 genes and not read the bold print. 23andMe actually tries to do a decent job on this, I think, that says we're only testing 3 of 1,000 dangerous versions of this gene. If you test negative, that doesn't mean you don't have it. But there'll be a lot of people who misunderstand that. So I think there are some real issues. Some of it is policy. Some of it ultimately is philosophical in the extent to which you are, where you come down on the spectrum between paternalism and free will, paternalism and liberty.
I think one question raised by this is not so much the personal idiosyncratic thing, but let's say we're talking about now careful experimentation, and you do get methods of doing this. Is this going to be available only to wealthy people initially, or people in wealthy countries and non-wealthy countries? A different kind of delivery, which is not particular to this, of course. This is just a matter of healthcare inequality anyway. But I think it's an issue to look at looking forward. Other questions? Yes.
Thank you. Aldous Lucas, Brooklyn College, CUNY, retired. I have a question to both, well, 3 questions actually, to both of you about innovation in general, which this is one example, but it's not quite as popular at the moment as, for example, social media innovation has been. First is, and it's a question basically about self-examination that I think scientific community hasn't been doing very much. First one is, we seem to be very biased as scientists with our new knowledge that we create all the time, but we ignore the fact that the ignorance about our material world is much, much bigger than the knowledge that we have produced in the last couple hundred years. An awful lot of knowledge, but nevertheless we live in a very complex material world. And how do we deal? I think that has something to do with how we deal with innovations. Second question is, techno-innovations have been largely done by physical scientists, but not by social sociology, human and social sciences. And I think innovations hit society, and that means society reacts to it. And for example, populism is largely anti-elitist. And I think it has something to do with the fact that we treat innovations as almost always good and not even pay attention to how they hit parts of our population. And the third one is scientists basically haven't played roles of advisors when it comes to regulation of innovations and so on. And they have not been either the legislators or the implementers of legislation to deal with it. I think that is not quite right. There should be many more scientists in Congress and also many more in the executive branch. I want your reactions.
Maybe it could be a reaction, but I'd like us to get to questions specifically having to do with CRISPR or this kind of thing, because we have here two experts, especially Jennifer, but also Hank, with having thought about these things, and just to get some. So I don't know if you want to respond to that briefly.
Sure. Democracy is the worst form of government except all the others we have tried from time to time. Any kind of governance is difficult. Humans are individually incredibly complicated. Collectively, we are complicated to the 7.5 billionth power. There are huge problems with limited education, limited understanding, limited desire to understand, magical thinking, desiring things to be the way you think they are rather than as they really are, and getting societies to listen. I think those are huge problems. I don't have any great solutions for them other than we keep plugging away with education and self-reflection and critical assessment of various innovations and their pluses and minuses. But I do think, as I said at the end, science lives in its cultures. And if you are unfortunate enough to live in South Dakota, which makes it a crime to do human embryonic stem cell research, and you want to do human embryonic stem cell research, you either have to change the legislature's mind or you need to move. So ultimately, science and society are inextricably intertwined.
Yes.
Hi, Michael Rand from Teachers College here at Columbia. I was curious if you had any suggestions on how to train people to deal with some of these ethical issues. So for example, maybe a case study. How do you have people in your lab who are training in this technology, et cetera. How do you talk to them about ethical issues or work with them so that when they leave your lab, they hopefully use the technology in proper ways and do experiments that are not problematic?
Yeah, great question. Two things that I do currently. One is that I tell every member of my lab that I think they need to spend some time figuring out how to explain in a sentence or two, without using any technical language, what they're doing and why it matters. And those, that brief kind of description would be the kind of thing you could use on an airplane, in an elevator, with your grandmother, with your kids, your neighbors. I think there's not enough effort made. I know I never received any training in my career until very, very recently about how to do that kind of thing. And I think it's valuable because right now in scientific training, as you probably know, we actually train students to get more and more and more specialized and expert in their field, and not to take a step back and think about how do I explain this problem I'm studying and why it matters to somebody that has no background in this field. So that's one thing. And the other thing I do is increasingly, because of all of the advances in the field of genome editing, in particular over the last few years, and for example, with the announcement last November about CRISPR babies, we've been having fairly regular meetings of not just my lab, but our Innovative Genomics Institute group at Berkeley and UCSF to talk about some of the bigger implications of the science that we're all engaged in. And that's been actually very interesting, and those meetings are very well attended. So there's lots of interest on the part of scientists. I don't think scientists are uninterested in these questions. They're just typically not encouraged or trained to talk about them.
And I think the training needs to go beyond just scientists and education of everybody, because we're all affected by these technologies. Education in high schools, education in college. It may be ethics classes, but you can also drop vignettes and case studies into substantive classes. We do some of that at Stanford and some of our biology and human biology classes. The key, I think, is not to try to teach answers and not to try to get them to read lots of Immanuel Kant, but it is to show them, take technical problems, and they can be in biology or neuroscience or cyber stuff, and make them think about how to think about it. What kinds of questions to ask? What should you be worried about in assessing a new technology? Ethically, socially, legally, politically, etc. And you can also do that through the media, and there are shows that do try to bring that sort of thing out. So education will never be the perfect answer to anything, but it'll be an essential part of the answer to everything.
I want to raise not really a question, because I think you both agree on this, but it's true certainly, as you say, that the human genome isn't fixed, that it's changing all the time. But we do want to be very careful about studies ahead of time, or knowing what we're doing and what those implications are. And of course, we can never really be sure, maybe never is not quite the right word, but I think not, down the line, what there are often unintended consequences. And so it's not that there's something sacred about where the genome is now, but still a lot of caution as we move ahead. And as both of you said, Jennifer said, the need to do research and to see what we know, but then to be very careful about any of this kind of thing.
I'm never going to argue against being careful,
Right.
except at the extreme. Not acting is also a form of action. And there are unintended consequences of
That's
not
true.
doing things.
Right.
There are kids
Right.
who will die from
make it-
disease who wouldn't have died disease because we took so long. Now, we need to take some time. There will always be some people who will be lost because something happens a little later because we were appropriately careful. But it's always a balance between that. And I do think people have this idea of nature, humanity, the world as stable and static. And so anything new moves it out of that safe position. But it's not stable and static, and particularly human culture in the last 300 years has been anything but stable and static. We're like a runner who stumbles and is desperately trying to keep from falling flat on our face. So it's not that we have the nirvana that any deviation from is necessarily bad. That's not saying we shouldn't be careful.
Right, right.
But we need to remember that being careful has its costs too. Being too careful has its costs.
Any comments? Yes.
Hi, my name is Leslie Goldman-Franco, and I'm a GSAS alum, so I'm not a scientist. And my question is about the unintended consequences. Can you explain what is the current status? Like, if you were to edit one gene, do you know what happens to how other genes relate to each other? Like, could there be potential problems with, if you take out, let's say, if somebody has Huntington's, by removing that one gene, do you know what happens to other combinations? Is that a clear question? Do you understand what I'm trying to get to?
Yeah, you're really asking about the question about genetic interactions. And,
yes.
As scientists, we know that when you make a genetic change in an organism in the laboratory that you're studying, the effects of that change are often profoundly affected by what we call the genotype of that organism. So you can have related, worms or fruit flies, for example, where a genetic change in one strain has a different effect than it does in another strain. And the reason is that they have different genetic makeups that affect the outcome of that mutation. And the same is almost certainly true in humans. So I think that's something very important to keep in mind as research moves forward with, frankly, any kind of human genome editing, but in particular, germline editing. And I think when I think about what's happening, what will happen in the future, I suspect that, and I won't put a time frame on this, it's certainly not going to happen very, very fast, but I think over time what will happen is that there will be increasing appreciation of how at least individual genetic changes are, affect people in the human population. Largely, it'll initially come from natural variants in the human population. A couple of them were already mentioned, like ApoE4, which is a gene that can predispose people to Alzheimer's disease. Many people in the natural human population have that allele of that particular type version of that gene, but they have it in the context of lots of other genes that are different. So we have pretty good information that regardless of what your other genetic makeup is, if you have ApoE4, you are likely to be at higher risk of getting Alzheimer's. And I think, for things like that, where we have pretty good evidence from the natural human population, those will be initially the targets of genome editing, because we can be relatively confident that those changes won't have some very, unexpected outcome when they're altered. What'll be a lot harder, right, is to make changes in genes where there isn't that kind of information and be able to understand or predict what the outcomes will be.
Yeah. First, being a GSA graduate shouldn't matter. My last bio class was in 10th grade, and we could barely spell DNA. It's not that hard to learn enough to really be an informed consumer. You don't need enough to work in Jennifer's. You don't need to know enough to be able to work in Jennifer's lab to be an informed consumer and citizen on this. And I highly encourage you to learn more. Those of you who aren't scientists, the biology is fascinating and wonderful, and there's some great science journalists and some great science books. Jennifer and Sam Sternberg, one of your new Columbia professors, have a nice book called Crack in Creation, A Crack in Creation, that you can and should learn from. But on the certainty, nothing in science is certain. Science is a process to limit uncertainty, to get closer to truth, but nothing in science is ever certain. But I do want to stress and even take another step on what Jennifer said. If you are going from a gene variant that is unusual and we know causes disease to the variant that is common and doesn't cause the disease, we're pretty confident that's a good thing. To change from the cystic fibrosis, from the delta F508 CF allele to a normal allele, we're pretty sure that's not going to cause other problems. We can't be certain, but we're pretty sure. To go from a common allele to a rare allele, a common version to a rare version, which Hoyle wanted to do with the CCR5 gene, from one that is ubiquitous, not just in humans, but in all mammals, to one that's found in a small percentage of people, that's iffier. And what he actually did to move from a common one to one never seen before in humans, that's when you have to be worried. So you can't ever be certain, but you can be less certain and much less certain.
Hi, Kyle Kanicki, Columbia Technology Ventures. I actually just got my PhD with Eric Green, who was a collaborator with Jennifer and Sam. My question is actually going back to the agricultural side, but it's certainly deep-rooted in the ethics and some of the things that we're discussing here right now. So I'm a pragmatic environmentalist, and I see certain things that are going on in the environment, especially declining fish stocks and a new salmon that's actually being genetically engineered coming out of Canada that has a single change that makes them grow faster, bigger, and holds great potential to actually solve a big problem in declining fish stocks. Now I also see that there's a lot of vitriol and vilification of those three letters of GMO, genetically modified organisms. And I want this fish to survive because I want salmon stocks to continue to rise. I'm also a scientist, and I believe in the free dissemination of information, but I understand how society can actually be very confused when it comes to some of these very complex genetic aspects. How do you feel about labeling these products with GMO that a lot of people are actually pushing for, saying, I want to know the information, I want to know whether this is GMO or not, but I would also more than likely result in these products failing?
I've got mixed feelings about it. I think if people want to know it, they should be able to find out. I do worry that in this case it may lead to more people rejecting something that is perfectly safe, healthy, and good for the environment, and all sorts of other good things. A great example, the Impossible Burger, invented by a biochemist at Stanford who gave up his job to push the Impossible Burger, completely plant-based burger that tastes like a hamburger. They're really good. Is being boycotted by PETA and Greenpeace because the heme in it comes from a transgene. It's a soybean gene moved into yeast to make the protein. That's crazy that you would oppose that. So the labeling does feed into some strains in our society that I think are going to misuse it. On the other hand, I think if people want the label, they're going to get the label, and we should encourage them to understand better what the label means and doesn't mean. California years ago passed a proposition that says you have to warn people if there's carcinogens in a location. And this was an environmental thing to try to get people to stop using carcinogens. But the result is every bar, every grocery store, every garage has this California warning, and everybody ignores it. And maybe that's what will happen with GM labeling. That's my best case.
Thank you.
Next.
Hi, I'm Abrianna Decker. I'm a grad student here and I did undergrad at Berkeley, so go Bears. So I know that he had previous mentors and collaborators back in America who knew the rough outline of what experiments he was doing, but took his word that he had local authorities and permission by bioethics committees from that hospital. So are there models being put forth for a middle ground between an international registry of every CRISPR experiment that we do versus trusting local authorities to make the right calls? Or is there anything that's already put forth or that's being debated, or what would you like to see in terms of that final model?
I think you raise a really interesting question and point, which is that, and I think many of us in the scientific community have been scratching our heads about this, is that suppose any of us knew that something like what Dr. He reported was going on prior to it being announced publicly. What if you knew months ago that something like that was happening? What would you do about it? Who would you tell? Who would you report it to? It's not clear to me how any of those scientists who were aware of it ahead of time could have stopped it from happening or even gone to an authority to report it. There isn't really an obvious path, I don't think, for doing that, although maybe Hank will disagree with me on this. But I think that now that this has all come out in public, I think there's a lot more open discussion about exactly that. How should it work? How should people be able to deal with information like that? And I think the announcement by the World Health Organization, this call for a registry, is an attempt to propose one path forward for this, which would be that there's transparency, that anyone working on something as ethically challenging as human embryo editing for clinical use should need to make that publicly known and report what they're doing, not hide it and do it in secret. So I think that is a potential path forward. I don't think we can have a registry for every CRISPR experiment. It's just not practical, but I think maybe for these certain types of applications that might make sense. And maybe another outcome of these international forums that are being convened will be to provide some kind of authority that could receive reports of these kinds of applications, even if they're not made public in a registry.
Yeah. So I think the registry, transparency of what's going on is important. But the issue of if you know what do you do, Matt Porteous, my Stanford colleague, who was one of the people in that position. I held a panel with Matt, and he said, you know, I wish I had told somebody. And I thought about it at the time, but I didn't know who to go to. I think we need two things. We need science to agree. If you see something unethical, and not just CRISPR, but you know, virus work, biological warfare work, flu work, there's some flu work that was controversial, you should report it to somebody. So create the expectation, the ethical aspiration, the ethical goal, that if you see something, you should say something, but then give them a mechanism by which to do it. And that's going to be tricky. Nobody likes snitching, or nobody likes snitches, and it's costly to inform on people. But we do have segments where we do it. So if you're a healthcare worker and you see bruises on a kid that are consistent with physical abuse, even if you don't actually think the parents were abusing, you're required, at least in California, to report that. And there are mechanisms to do that. If you're a doctor in California and you've got a colleague who is impaired by drugs or alcohol, you're required, to the extent where you think it's a safety issue for her patients, you're required to report that, and there's a mechanism that allows that, that's made it a much more common. I think science needs something like that.
Thank you.
Hi, my name is Emma Kenny-Passia, and I'm a senior here at Barnard College. Thank you so much for this talk. It was so interesting. My question is surrounding Dr. He's experiment again. Obviously, there were a whole host of ethical issues that you touched on surrounding it, but one of the more interesting spheres of discourse, I guess, at least to me, after this experiment was his choice to edit the gene associated with HIV particularly. Obviously, super stigmatized disease still, and we do have treatments today that allow HIV positive people to live really long, fulfilling, and like mostly healthy lives. So I guess my question is, with these technologies that offer the promise of possibly like curing or treating diseases genetically, how do we balance that with existing treatments that treat these diseases non-genetically? And similarly, I guess more from a policy perspective, like a few years ago when Obama launched the Precision Medicine Initiative, I think there was a lot of concern after that it might draw funding from, for example, like social interventions to mitigate social determinants of health, rather than like immediately going to a genetic solution. So I'm wondering how policy-wise or just ethically we balance those kind of two solutions.
Well, I could take the first one, maybe Hank will take the second one, but for your first question, you know, how do we balance genetic versus non-genetic treatments for disease? To me, I think it always comes down to risk-benefit analyses, right? So you have to ask yourself, first of all, how severe is the situation, whether it's a disease or a predisposition to disease, versus the treatment that you could offer to people. And in the example of the CCR5 gene that we talked about with He Jianqui's work, there, you know, as maybe you know, there are well-established procedures to help parents who have active HIV infection to have children that are not, first of all, not infected by HIV, but secondly, as you pointed out, people that, you know, there are well-established procedures to avoid getting infected by HIV, even if you have a active CCR5 gene, and also if you do get infected with HIV, to control that infection and live a largely normal life at this point. In China, so when I met with He Jianqui, he was very passionate. He said to me, Jennifer, you don't understand. You come from America where things are different. But he said in China, there's an absolute stigma against HIV infection and infected people. These people live under a social stigma for their entire lives. And what I was trying to do was to give these parents and their kids an opportunity to get out from under that kind of stigma. Now, was he sincere in that desire? I can't say, but he was certainly passionate in expressing that desire. So, you know, I do think we have to be sensitive to, you know, the kind of the different kinds of impacts that disease can have in different parts of the world, and HIV is one that is associated with a certain social kind of response. What's the right answer in this case? I certainly think what he did should not have been done, and was clearly unethical. But going forward, I think it's going to become increasingly, I think, as Hank said earlier, there's not really a clear line between what's medically necessary or not. And as the technology for human embryo editing continues to advance, as it assuredly will, I think, we're no longer going to be able to say, well, it's the technology isn't there yet. It will be there. And then we really have to ask ourselves these more, nebulous kinds of questions about using it.
Thank you.
So on the He point in particular, there's a more nefarious explanation as well. You can save the children from the possibility of prenatal infection from a father's HIV infection by an easy process called sperm washing. HIV doesn't infect the sperm, but the particles can be in the seminal fluid. So you wash the seminal fluid away, you use the sperm. The problem in China is that HIV people aren't allowed to use assisted reproduction at all, including sperm washing. So one theory is that He picked this in part because he had people who desperately wanted to have non-infected children who couldn't legally get it in China, and he was offering them not only a way to do that, but a free way to do that. The press release that is our only version of the one investigative report so far apparently done in China says that, among other things, He paid people to take blood tests at the IVF clinic who were HIV negative to pretend to be the fathers, because if the IVF clinic, they were required to test it. If they tested positive, they couldn't take on that parent as a patient. So there's a lot of potential coercion in that. Your broader question, though, about how we pick what we go for, it should be risk-benefit and risk and cost-benefit, but it often isn't, because we like hot, sexy things. The news does, we do, Twitter does, the stock market does, and that's something we constantly need to fight about, to fight back against. The hottest, sexiest thing might be the best way, but it might just be eat less and exercise more.
Thank you so much.
One more question, because you'd asked before, but then we should stop, I think. And go. Yeah, sure, go ahead.
I don't have an affiliation. I'm just a curious interloper. So you guys have talked about how Dr. He's experiment was logistically improper, but and you've also mentioned this, the axes of therapy versus enhancement, and preventing neonatal HIV infection would, I think everyone would agree that's a therapeutic, and that what he violated was, sort of bioethics, not testing this first outside of humans, et cetera, informed consent. But also, in China, homosexuality is illegal, right? This may also be considered therapy there, and they may say, okay, to avoid international outcry, we will logistically proceed in an ethical manner so that the experiment is, done with informed consent and, experimented outside of humans first to make sure it's safe, et cetera. How do we prevent, because every country, government, society, whatever, is going to have different social mores about, and there's even more gray areas like, Asperger's syndrome, sociopathy, things that may not be, they may be considered therapeutic by some cultures to eliminate, sort of Minority Report style. Like, how do we prevent or police that, and should we?
Okay, it's a big question.
It's a big and deep question. First, the good news is, at least in the CRISPR context, we don't know genes for sexual preference. We don't know genes for autism or neurodiversity. We don't know genes for a lot of these things yet. We may, or it may turn out that they're too complicated and we may never know it. But the deeper question, and this is one of the deepest questions, I think, in all of policy and ethics, are there universal human rights, or is it all culturally relative? And if it's all culturally relative, then if China's culture says this is the way it should be, can outsiders say, no, it isn't? And that seems wrong. On the other hand, if there are universal human rights, what are they and where do they come from? And how do you define them? In which mountain did they come down on which stone tablets? And to me, that's one of those questions that's above my pay grade. I'm just a pragmatic law professor. I just try to do what I can. But that is maybe the deepest, hardest question in policy and ethics, cultural relativism versus some universal ethics. And I don't have the answer.
It's a question I could speak on, but no, I think we have to stop here. So thank you very much, both of you. I think this has been